electron transport chain. All right. All right. And what we want to say about this that's really important is that it is where we have oxidative phosphorylation. Okay. We also want to say that it requires oxygen and that it is happening in the mitochondria. Okay. So just to illustrate exactly where this is, if you remember, think back to the structure of the mitochondria. This is what it looks like, right? It's got this highly folded membrane, which separates it into an outer layer. So this is the outer mitochondrial membrane. And this would be considered the inner mitochondrial membrane. All right. And where the electron transport chain is located and where oxidative phosphorylation is taking place is right across this surface, so across the inner mitochondrial membrane. Okay. So we'll put a little star here so that we know exactly where we're going to be zooming in on. Okay, so let's draw that. So here's our outer mitochondrial membrane. Here is our inner mitochondrial membrane. All right, this space here is called the intermembrane space. All right, and we also want to describe what's happening as far as the various gradients that we've created here. So all of the electrons that we have been describing, they're going to be shuttled off here to the mitochondria, and they're going to be in higher concentration in the intermembrane space. Okay, so let's draw a lot of electrons permeating this space, because this is where we're going to end up having them in a higher concentration. All right, and let's put that there is a high total concentration of our electrons up here. Okay, and let's contrast that with the actual mitochondrial matrix. All right, so let's label this space out here. This is going to be considered the mitochondrial matrix, which is in here, right? So this central space. So this is the mitochondrial matrix. And by contrast, we're going to have significantly fewer electrons in the mitochondrial matrix. So we'll draw a sparse view down here. And we're going to mention here that it's going to be a lower concentration of the electrons. Okay. All right, hopefully everyone is with me so far. Let's begin to put our transporters in. So the reason that we're calling this the electron transport chain is that electrons are going to be transported through a series of transporters that are inserted in the inner mitochondrial membrane. So we've got transporter 1 here. Okay. And this is really a channel, so let's kind of draw this opening here in the center to represent that. All right, we've got transporter 2 here. Three here. Okay. And then the fourth one is the most important of them all. This one is called ATP synthase. And I'm going to draw it in green because it's going to be moving along a different direction. Right? Okay. And this guy is called ATP synthase. Why? Because it's going to be the most important part of making our ATP. So really important to remember the name of this transporter. These other transporters are also uniquely named. I'm not super concerned for you guys to know the names of these. That's not really important for the scope of this class, but I do want you to know the name of the fourth one, which is going to have a really pivotal role in making ATP. So the ATP synthase is the final transporter of electrons that's inserted in the membrane here. All right. Now, what ends up happening is that we're bringing in these electrons. So all of the electrons that we described as coming from glycolysis, from the Krebs cycle, from that linking step, they're going to come into the mitochondrial matrix. All right. And they're going to start moving through they're going to start moving across the membrane. So they're going to enter here. So let's kind of draw this out and talk about how these are converted. So we had NADH, H plus. Remember, this was the reduced form that was carrying electrons. That is then going to be oxidized here in the matrix to give us NAD plus. And then we're going to liberate some of those electrons. Okay. So remember, this is coming from glycolysis. So we talked about these coming here, and this is what their purpose is when they are in the mitochondrial matrix. Same thing with FAD. We talked about FAD, H2 being liberated from the Krebs cycle. That is also going to be oxidized to give us FAD. And we're going to free up these electrons. So these electrons are really what we need to drive this transport chain. All right. So what's happening here is oxidation, just to remind ourselves that when we are gaining electrons, right, liberating or freeing them as we are here, this is called oxidation. When we were making them back in glycolysis and the Krebs cycle, that was called reduction, okay? So we put these electrons into this mitochondrial membrane. And as we move hydrogen ions, so we have lots of hydrogen ions here, very few hydrogen ions here, that creates a gradient, right? When we have the movement of hydrogen ions going across the inner mitochondrial membrane against their gradient, that's going to require energy, and that is what we're using the electrons to do. So the electrons are giving us the energy, right? That's why these are shown in red, because they're going to use the energy of the electrons. And as we pass the electron from this transporter to this transporter, we take that energy and move hydrogen ions from a place of low concentration to a place of high concentration, and that's why we end up having much more of our hydrogen ions in the intermembrane space as compared to the mitochondrial matrix, okay? So we, again, move the electrons from this transporter to this transporter, and we pump some more hydrogen ions in this direction. Same thing here, we move some electrons from this transporter, the second one to the third one, and we liberate the energy to then move the hydrogen ions in this direction, okay? And I really want to illustrate this well, so I'm going to put the actual hydrogen ions right here, and then them being liberated on the other side with the energy. So I'm going to put this arrow here in green as well, with the energy that was taken from the movement of the electrons, right? So we can begin to see how the name makes sense, right? It's the electron transport chain. Electrons are being passed from this transporter to this transporter to this transporter, and that is taking the hydrogen ions in this area where they're already in a lower concentration, moving them against their gradient. And whenever we move something against its gradient, that takes energy. And that is the purpose of the electrons, to give us the energy that's required to move these hydrogen ions against their natural gradient. Now, what that does is it creates this high concentration of hydrogen ions up here and much lower concentration down here. And that has a lot of potential. We'll talk about membrane potentials in our next chapter coming up. But whenever we have a large separation of charge, a lot on one side and a little bit on the other side, that is potential. That is energy that can be harvested to do a lot of work. In this case, that energy is going to be used to make ATP. So we're going to take all these hydrogen ions, they're going to be moved along the final transporter, which is the ATP synthase. And that is how we make ATP. So the movement of all these hydrogen ions through the final transporter, which is the ATP synthase, is what will yield the maximum amount of ATP. Okay, that's why we call this transporter the ATP synthase, because it's going to synthesize ATP. It's going to take the ADP that we have in the cell. So it's going to take the ADP that we have in the cell here. And then the energy that's yielded here is going to add a phosphate group onto the ADP, and we're going to yield ATP. Now, the amount of ATP that we yield is going to be what is probably the most important part here. So we're going to yield 34 ATP per glucose. Okay, so for all of that ATP that we're yielding here, it's only coming from the breakdown of a single molecule of glucose. It's going to start in glycolysis, continue on into the linking step, continue on into the Krebs cycle, and all through those initial stages, we're making the electrons that we need to really drive this train. We're also making some of the additional byproducts that we're going to need to drive this chain as well. And then what's happening here is really just the last piece of the puzzle, where we create this gradient with the help of the electrons, and then we move these hydrogen ions down this final transporter and make a really large amount of ATP. Okay, so we're going to make 34 ATP for a single molecule of glucose. All right. Now, where are those electrons going to go? Who is the final acceptor of these electrons? This is where oxygen comes into play. Okay, this is why we need oxygen. So oxygen is standing by here. All right, here's our oxygen. And it is going to be the final electron acceptor, because if we didn't have oxygen, guess what? This whole chain would halt. We need someone or something to be accepting the electrons so that the chain can continue passing more electrons along, so that we can continue making more of this gradient, so that we can continue making ATP. So oxygen is really important. It's going to be the final acceptor of the electrons to drive this process continually so that we can make a lot of ATP. All right, so let's make a note here that oxygen is the final electron acceptor. All right, this guy. Okay, I'm going to pause here and see what questions we've got so far. All right. Okay, do you guys need a minute to finish up drawing? I'll give another minute just in case you were drawing along. And then we'll go ahead and sort of recap and wrap up. Okay. And I will post these as separate drawings on Brightspace as well, but only in case you missed something. It's really important for you to still draw these out in class, right, with me. So let's go ahead and clear this. Okay. So just to recap what we've said in that drawing, we're going to go through the schematic that's shown here. So the donation of electrons to the electron transport chain is happening via the oxidation of NADH to NAD+, and the electrons are now liberated here. All right, so we've got 2 electrons here, 2 electrons also liberated from FADH2. So if we oxidize FADH2, we're going to get back FAD, and they want to liberate these 2 electrons, which is sort of hitched on to our FAD. And then the final accepting of electrons is happening by the presence of oxygen. So oxygen is going to grab those electrons as well as the 2 hydrogen ions, and that is how we make water as a byproduct of this process. Okay, the electrons along with hydrogen ions and oxygen will make H2O, which is going to be water. Okay, so just recapping what's happening on this side: glycolysis, the linking step, the Krebs cycle. They're making 10 NADH plus, 2 FADH2. All of that is being shuttled off to the mitochondria for oxidative phosphorylation. We're going to invest oxygen, right? Remember I said this is the whole point of us breathing oxygen in, is for it to be present here to accept those electrons. We're also going to invest 28 ADP as well as 34 phosphate. And then we're going to yield on the other end NAD+, so this is in its oxidized form, FAD also in its oxidized form because it has now liberated those electrons, and then water as that byproduct, as well as 28 ATP. Now you can see a variation in how much ATP is made. This varies just from textbook to textbook, from source to source. I'm going to encourage you to stick with 34 ATP per molecule of glucose. That's what I'll expect you to know on your test. But you can see as few as 28 ATP or as much as 38 ATP in some sources, depending on a lot of different factors: age, gender, your body composition, that can change how efficient your body is at using glucose and how much or how little ATP you make per molecule of glucose. Okay. All right. So this is sort of what we drew out initially. So showing you where that's happening. Here's our inner mitochondrial membrane. Here is our intermembrane space. Here is the matrix. Here is the oxidation happening, which is liberating the electrons that we're using. We're passing them from this first transporter or complex, the second transporter or complex, on and on across the chain. And then what that is doing is that is allowing us to move hydrogen ions from a place of low concentration to a place of high concentration. The red arrows here mean that they are moving against their concentration gradient. So typically, molecules like to move along their gradient, so from high to low. So in order to move them against their gradient, right, we're going to have to put this energy in, and that is what the electrons are doing. The final transporter, which is ATP synthase, can then move these electrons down their gradient, or these hydrogen ions down their gradient, and that can be used to phosphorylate ADP and make ATP. Meanwhile, oxygen is standing by to be the final acceptor of the electrons at the very end of the chain. It's going to make water as those electrons are combined with hydrogen ions and oxygen, and then that will be released as a byproduct.